So, hi everyone. So, I am Angeline Dayala. So, I will be discussing about the cell cycle that is focuses on mitosis and meiosis. Okay, so let's start. So, what is cell cycle? It is the ordered sequence of events that occur in a cell in preparation for cell division. The cell cycle is a four-stage process in which the cell increases in size. GAP1 or G1 stage copies its DNA. The synthesis or S stage prepares to divide, or G2 or GAP2 stage, and divides the cell, so mitosis or the M stage. The stages G1, the synthesis, and G2 make up interphase, which accounts for the span between cell division on the basis of the stimulatory and inhibitory messages. A cell receives it, decides whether it should enter the cell cycle and divide. So let's go to the stages or the phases of the cell cycle. So here, GAP1 or G1, cells increase in size in G1, produce RNA and synthesize protein. An important cell cycle control mechanism activated during this period, the G1 checkpoint, ensures that everything is ready for DNA synthesis. So in this part, it will proceed to the S phase or the synthesis phase. It is the phase of the cell cycle in which DNA is replicated, occurring between G1 phase and G2 phase. Since accurate duplication of the genome is critical to successful cell division, the processes that occur during S phase is tightly regulated and widely conserved. Then the next one is the G2 phase or GAP2 phase or growth 2 phase. Is the third subphase of interphase in the cell cycle directly preceding mitosis. It follows the successful completion of S phase, during which the cell's DNA is replicated. So here is what is mitosis. So mitosis is a type of cell division in which one cell, which is the mother, divides to produce two new cells, what we call the daughters. What are the genetically identical to itself? That are genetically identical to itself. In the context of the cell cycle, mitosis is the part of the division process in which the DNA of the cell's nucleus is split into two equal sets of chromosomes. So here is the example of mitosis. So we have here the stages of the mitosis, which are the prophase, metaphase, anaphase, and cytokinesis and telophase. Okay. So next is the phases of mitosis: prophase, metaphase, anaphase, and telophase. Cytokinesis typically overlaps with anaphase and or telophase. The most important to understand is what's happening at each stage and why is important for the division of chromosomes. In early prophase, the cell starts to break down some structure and build others up, setting the stage for division of the chromosomes. The chromosomes start to condense, making them easier to pull apart later on. The mitotic spindle begins to form. The spindle is a structure made of microtubules, strong fibers that are part of the cell's skeleton. It's a job to organize the chromosomes and move them around during mitosis. The spindle grows between the chromosomes. As they move apart, the nucleus or nuclei in plural, a part of the nucleus or ribosomes are made disappear. This is a sign that the nucleus is getting ready to break down. Okay, so here it is. So this is the mitotic spindle. This one that starts to form. Then this is the chromosome starts to condense. Nucleolus is gone. Okay, so let's go to late prophase. In late prophase, also called chromatophase, the mitotic spindle begins to capture and organize the chromosomes. The chromosomes become even more condensed, so they are very compact. The nuclear envelope breaks down, releasing the chromosomes. The mitotic spindle grows more, and some of the microtubules start to capture chromosomes. In metaphase, before that, so here it is. So the spindle that last was only developing, so it catches the chromosomes, breaks down. Okay, so let's go to the phase. Metaphase rather. So in metaphase, the spindle has captured all the chromosomes and lined them up in the middle of the cell ready to divide. So here it is. This chromosomes lined up at metaphase plate. Okay, so next one is the anaphase. The sister chromatids separate from each other and are pulled towards opposite ends of the cell. As you can see, they're like separated from each sister chromatids. Okay, so let's proceed to the telophase. The cell is nearly done dividing and it starts to reestablish its normal structure as cytokinesis or division of the cell contents takes place. Here it is how they divided. So here is the start of the chromosomes to decondense. Then the spindle disappears. Then the nuclear membrane reforms, the nucleus reappears. Okay, as you can see, the labeled part. So here is the cytokinesis, the division of the cytoplasm to form two new cells overlap with the final stage of mitosis. So this is an example of the animal cell that is performing cytokinesis. So actin ring and the cleavage furrow. So here's the plant cell, cell plate, and cell wall. Okay, so in animal cells, cytokinesis is contractile, pinching the cell into like a coin pores with a dough string, and the dough string is a band of filaments made of a protein called actin, and the pinch crease is known as the cleavage furrow. Plant cells can't be divided like this because they have a cell wall and are too stiff. Instead, a structure called a cell plate forms down the middle of the cell, splitting it into two daughter cells separated by the wall, as what you have observed this. Okay, so that's it for the mitosis. So let's go to meiosis. So what is meiosis? Meiosis is a process where a single cell divides twice to produce four cells containing half the original amount of genetic information. During meiosis, one cell divides twice to form four daughter cells. So meiosis can be divided into 9 stages. These are divided between the first time the cell divides in meiosis 1 and the second time it divides in meiosis 2. So let's proceed to meiosis 1. Interphase. The DNA in the cell is copied, resulting in two identical full sets of chromosomes. In phase 1, the copied chromosomes condense into X-shaped structure that can be easily seen under a microscope. Then, metaphase 1, the chromosomes pairs line up next to each other along the century or the equator of the cell. Then, the fourth was the anaphase 1. The pair of chromosomes are then pulled apart by the mitotic spindle, which pulls one chromosome to one pole of the cell and the other chromosomes to the opposite pole. The fifth one was the telophase and cytokinesis. The chromosomes complete their move to the opposite poles of the cell. So in meiosis 2, prophase 2, now there are two daughter cells, each with 23 chromosomes or 23 pairs of chromatids. 7 is metaphase 2. In each of two daughter cells, the chromosomes pair of sister chromatids line up end to end along the equator of the cell. Then the anaphase 2, the sister chromatids are then pulled to opposite poles due to the action of the mitotic spindle. So the last one is the telophase 2 and cytokinesis. The chromosomes complete their move to the opposite poles of the cell. At each pole of the cell, a full set of chromosomes gathered together. A membrane forms around each set of chromosomes to create new set of nuclei. So here is the example. As you can see in the interphase, there are centrosome and the chromosomes. So when the DNA copied, so we proceed to prophase 1 where the chromosomes paired up. Then the recombination occurs in metaphase 1. So the chromosomes lined up the equator of mitotic spindle, just like the last one I've mentioned. So then the next one is the anaphase 1, where the chromosomes pulled apart. Then the telophase 1 and cytokinesis, cell pinches in the middle. Okay. Then in prophase 2, here is the two daughter cells. Then the metaphase 2, chromosomes lined up in an equator. In anaphase 2, sister chromatids pulled apart. Then the telophase 2, cell pinches in the middle. Then the four daughter cells are made. So that's it. So we have here three So in the zygotic meiosis, life cycle of protists is of two types. One is the cell cycle showing zygotic meiosis. Two is life cycle showing gametic meiosis. We focus on life cycle showing zygotic meiosis. When protist is haploid and meiosis occurs in zygote, then it's known as zygotic meiosis. In this type of life cycle during sexual reproduction, gametes are formed by mitosis. Note, in this life cycle, zygotic phase is only diploid and remaining other phases are haploid. So this type of cycle known as haplotonic life cycle. So here is the example. Okay. Can you see that? So it's also called, the zygotic meiosis is also called haplotonic life cycle. Okay, so that's all, and thank you everyone for listening, and I hope you learned about my discussion, and sorry for some noises that you have heard in the background since I'm here at my house, so you will also heard some noises around. So thank you and God bless.